Matt Ridley, Genome, the Autobiography of a Species in 23 chapters. In A Genome, the Autobiography of a Species in 23 chapters, Matt Ridley dives into the fascinating world of the human genome and its impact on our lives. Entering the world of genetics and biology, the book unravels the complexities behind our genes and how they shape our physical, psychological, and emotional attributes. Embark on this enlightening journey as we explore intriguing themes such as genetic development, adaptation, aging, and gene-linked diseases. By the end of this summary, you will have a deeper understanding of the role of genetics in human evolution and a newfound appreciation for the book that is our genome.
Unraveling the Human Genome. The groundbreaking moment when British Prime Minister Tony Blair and American President Bill Clinton announced the rough mapping of the human genome on June 26, 2000, marked a historic turning point. This breakthrough granted us a peek into the book that narrates the story of human life. The human genome is composed of 23 chromosome pairs containing genes that reside on strands of DNA. These strands are made up of nucleotides, adenine, cytosine, guanine, and thymine, which are crucial in replicating the genome. With over 1 billion words, the complex and information-rich human genome represents the evolving story of life from bacteria, fish, and apes to humans. It is within the genome that we can uncover how various illnesses emerge and how individual reactions to everyday experiences come into play. Our genetic connection to nature. While we may assume that we, as humans, have evolved beyond other species, our genetic makeup suggests otherwise. Sharing most of our genes with other animals, humans and chimpanzees have only a 2% genetic difference, with one less pair of chromosomes being a key distinction. As descendants from a common ancestral ape group, humans and chimpanzees followed separate evolutionary paths, ultimately resulting in Homo sapiens. Although humans have experienced remarkable adaptation, our longer lifespans make genetic development slower compared to bacteria, which can adapt rapidly due to shorter lifespans and more frequent generational changes. Despite the belief that humans are the pinnacle of natural selection, our similarities to other species challenge this assumption. Genetics reveal that we share the majority of our genetic material with other living organisms, establishing an undeniable connection between humans and the rest of the natural world. Take chimpanzees, for instance. With only a mere 2% difference in genetic makeup, our closest relatives exhibit striking similarities to us. The visible variations, such as our larger brains and reduced body hair, compose only a small fragment of our entire genetic blueprint. Perhaps the most significant distinction lies in our chromosomes. Humans possess one fewer pair than chimpanzees. So, how did our closely related genetic composition come to be? Millions of years ago, a single group of apes likely split into separate evolutionary branches, resulting in today's chimpanzees and, eventually, Homo sapiens. Adaptation played a critical role in this process, allowing each generation to refine its ability to cope with changing conditions. Though humans have come a long way in the evolutionary journey, our rate of adaptation pales in comparison to organisms like bacteria. Due to our extended lifespans, humans require more time for genetic development to occur. Bacteria, however, can achieve the same number of generations in just 25 years, making them highly adaptable in the face of an ever-changing environment. The tiniest inch, life or death. It's astounding how something so minuscule, like a tiny fraction of the human genome, can determine the fate of your life. Genes don't just affect appearances, they can determine how long we live. Take, for example, Huntington's disease, a fatal genetic disorder that causes the progressive breakdown of nerve cells in the brain. While everyone carries the gene, having more than 39 repetitions of the CAG genetic word, cytosine, adenine, guanine, results in the disease. Now, let's imagine stretching our genome to span the Earth's equator. That 1-inch section dictates life or a slow, agonizing death. Huntington's, however, is not the only disease caused by this CAG repetition. Scientists know of 6 such diseases, and mutations due to repeating genome sequences are especially concerning. As a genome replicates more frequently, genes can accidentally repeat sequences, causing devastating mutations. Unfortunately, few people choose to get officially diagnosed with Huntington's. After all, what can one do when discovering their predetermined fate? The microscopic nature of our genes and their immense power over our lives serves as a reminder that even the tiniest part of us can have an enormous impact on our existence. The paradox of junk DNA. Your genome, the blueprint defining your uniqueness, is filled with an unexpected abundance of seemingly useless genetic material. This so-called junk DNA was initially viewed as insignificant. However, researchers have found that only about 3% of our genes serve a purpose. Interestingly, some of this genetic garbage originated from ancient viruses that our ancestors' bodies tried to suppress. Repetition of certain sequences in junk DNA can generate abnormalities, including tumors and detrimental impacts on health. On the flip side, these repetitive patterns can also be helpful, as in DNA fingerprinting, which exploits the unique repetitions within junk DNA to set individuals apart. With such complex implications, junk DNA sparks intrigue and raises the question, what other applications might it hold for us? Beyond genetics, personality formation. Our personalities are not solely determined by our genomes. While genes play a significant role, key factors such as brain chemistry and environmental influences also contribute to shaping our unique characteristics. A gene present on chromosome 11, for instance, impacts dopamine receptors, thereby affecting an individual's decision-making and potentially having more severe consequences like Parkinson's and schizophrenia. Hundreds of genes play a part in defining our inherent traits. Nevertheless, environmental factors, such as upbringing and peer influence, play a crucial role in how our personalities ultimately unfold. Recognizing and accepting the innate aspects of our personalities can help us work on self-improvement without feeling embarrassed or distressed about our peculiar tendencies. Unraveling the aging process. Aging is an inevitable part of life, intricately linked to natural selection and the need to protect and nurture our offspring. The process really kicks in around age 55, when our children are typically self-sufficient and the older generation's purpose has been served. Aging is connected to the gradual depletion of telomeres, which protect chromosomes from errors as they duplicate. While many species possess genes that produce telomerase, a telomere-preserving enzyme, human cells lack these, leaving us susceptible to aging in conditions such as cancer. Cancer cells, possessing the elusive telomerase, remain immortal and continue to multiply without ever aging. Harnessing genes to cure disease. Genes not only cause diseases but can be the key to curing them as well. Gene therapy introduces helpful genes into diseased cells, stopping their destructive effects. Although most gene therapy research focuses on cancer, the ethical and moral implications of genetic modification raise concerns. Society must weigh the potential benefits of modifying humans against possible consequences and decide whether this is a path we should pursue. Our understanding of genes has come a long way, revealing their role in causing deadly diseases like cancer and Huntington's disease. However, the power of genes goes beyond triggering diseases. They can also be harnessed to cure them, thanks to genetic modification and gene therapy. Gene therapy is an innovative approach that takes the genes necessary to combat a particular disease and introduces them into the diseased cells. Essentially, this halts the progression of the illness, offering new hope for patients battling aggressive diseases. Cancer has been the primary focus of gene therapy research due to its devastating impact and ubiquitous nature. Every minute, someone in the United States succumbs to this lethal disease. Although gene therapy has demonstrated success in human experiments, its pursuit also comes with drawbacks, including ethical and moral dilemmas. Genetic modification has long been met with apprehension and even fear, with some drawing parallels to the unnatural creation of Frankenstein's monster. Despite these reservations, at the time of the book's publication, genetically modified, GM, crops were projected to make up 50 to 60% of all U.S. crops by the year 2000. The genetic engineering of animals for increased production has further intensified the debate, as society grapples with the possibility of extending these practices to humans. While genetically engineered humans remain an idea for the future, it is crucial to deliberate on what this would mean for humanity as a whole. Gene therapy and genetic modification hold the potential to cure genetic diseases in the future. However, society must contend with the profound question, should we take this path? The answer lies in carefully considering the ramifications and ethical concerns, a conversation that can shape our destiny. Unraveling the eugenics legacy. The concept of eugenics, or the breeding of better humans, dates back to 1885, far earlier than Nazi Germany's infamous implementation during World War II. In the early 20th century, American scientists campaigned to isolate bad stock from society, resulting in forced sterilizations of those deemed mentally unfit. Notable figures like H.G. Wells, Theodore Roosevelt, and Winston Churchill supported eugenics, with Churchill attempting and failing to pass eugenics laws in Britain. Although eugenics has left a dark legacy, today's ethical debates surrounding prenatal testing differ in that parents now have the choice to continue or end a pregnancy, rather than face government-imposed restrictions on child rearing. Free will versus genetic determinism. When pondering the concept of free will, we often wonder whether our thoughts and actions are determined by our genetic makeup or shaped by society. Many people find comfort in social determinism, attributing life choices to past and present environments. This mindset, however, may also lead to the illusion of free will. Interestingly, most people reject the idea of biological determinism while embracing social determinism. Recognizing the role of both genes and society in shaping our behavior can help us accept ourselves and make conscious choices in life. Are you truly in control of your thoughts and actions? Does your genetic makeup determine your choices, or do societal influences dictate your decisions? This age-old question will undoubtedly continue to stir the human mind for generations to come. Is free will real? Admittedly, it's hard to accept the idea that our entire existence might be controlled by our genes. The notion of biological determinism, which suggests that our actions are predetermined by our genetic makeup, makes us uncomfortable. We prefer the idea of social determinism, believing that events such as divorce, child abuse, or alcoholism are products of our past experiences and environments. For example, in social determinism, children of divorcees are more likely to follow suit due to their developmental exposure. But what if societal influences on our character only make our sense of free will an illusion? Unsurprisingly, people generally embrace the concept of society shaping our decisions but neglect the impact of genetics. By acknowledging that both genetics and societal factors play a role in shaping our behavior, we can better accept who we are. Though biological determinism might exist, it doesn't imply an unchangeable destiny. You still have the power to make choices, even if your genetic predispositions say otherwise. However, when we choose to focus only on social determinism, we run the risk of surrendering our free will and avoiding responsibility for our actions. It's peculiar how readily people accept society's control over their lives, yet vehemently oppose the idea that genes also significantly influence human behavior.
Matt Ridley's Genome offers an insightful look into the world of genetics, revealing how the human genome not only defines our physical characteristics but also plays a significant role in our personality traits and predispositions. This eye-opening exploration of our genetic makeup sheds light on the inner workings of our cells, the marvels of adaptation, the effects of aging, the potential for gene therapy, and the contentious topic of eugenics. By understanding our genome and its diverse applications, we can better grasp our evolutionary history and begin to comprehend the intricate balance between our genetics and the environment, thus opening the door to a future where genetic knowledge can potentially uplift the human condition.